Okay. And I just wanted to make a few quick comments to thank all the speakers and all the attendees for really, I found the talks riveting, really just amazing. Every single one was just great. We had an excellent Q&A session and all the moderators really kept us on time. So I wanted to extend my thanks and appreciation again to everybody, especially the speakers. It's Friday afternoon. Some people, it's real late. So, we want to keep on schedule. And with those comments, I want to introduce the panel, which is a distinguished panel of experts from earlier today. And that's going to be David Rosenblatt, Harvey Levy, Martina Humer, and Marzia Pasquale. And we're going to have a panel discussion, which I'm going to moderate. And so what I wanted to do was to start off with sort of more broad, open-ended questions to provide some perspectives to all of us. And for that reason, I wanted to start off with, actually, I'm going to call on people to make this, so there's no pauses. I wanted to start off with Professor Harvey Levy and ask you, Harvey, to share with us, your perspectives on how things have come since the early days. And you've been such a pioneer, I know we've had our conversations and I remember them vividly about, being involved in the earliest days of newborn screening and sort of, how things are going from the perspective of taking care of the patients and new therapies. And, what do you think the future holds for us? And if you could share your perspectives in, maybe a couple minutes, that would be great. Then I'm going to ask Dr. Rosenblatt the exact same question.
Well, let me just start by telling you that we've come half a world or maybe 3/4 of a world from the time when homocystinuria was first identified in 1962, as we've said, and where we are today. When it was first identified in 62, there was no therapy. And as a matter of fact, quite a number of years there was no therapy. There's low methionine, soy formulas, which are low in methionine, and they were used. Tom Perry, for instance, in Vancouver, was a proponent of that, but that didn't work very well. So it didn't really get the homocysteine level down. And as a matter of fact, we weren't measuring total homocysteine at that time. We only measure homocysteine in the last 20, 25, 23 years. So quite frankly, we were looking at homocystinuria sort of a gross way rather than the more microscopic way that I think we can look at it today. So we've come a huge way. And of course, naturally, we've come all the way up to enzyme assays and then identifying the gene and characterizing the gene and mutations. And now we're getting into basically be getting into enzyme replacement therapies. And we're talking about changes, let's say, short of replacement therapies with various metabolites. And now we're getting into, we're talking about gene therapy. So we've come a huge way from the very beginning all the way up to the present time. But it was rough going early on, and I can tell you that the going now is a whole lot smoother.
So thank you. My connection cut off for just a second, but I wanted to ask you, maybe just to finish the comments, do you think we're going to be approaching an era where we have effective newborn screening, find our patients early, and they're going to have, almost normal outcomes? Or do you still think we have a ways to go?
Sure, we have a way to go, and I think the answer is yeah. Eventually, I think we will get it to the point where we'll be identifying every baby with homocystinuria or the Kamahlman defects in the newborn period, and we'll probably have much better therapies all the way down the line, which are going to optimize clinical outcome. To the extent that they'll be optimal is another question. Will they be totally normal? Sure. I mean, eventually, I think we will develop therapies. But when that's going to be is anybody's guess.
Well, that's an amazingly positive comment, I think, for all of us. And certainly, I think we all feel the enthusiasm excitement about the new therapies and the combined efforts that we're making on all fronts.
Sure, Chuck. When you and I first started talking about this, it was a different ball game, as a matter of fact, and that wasn't the very beginning.
Yeah.
So we've come a long way from the time that we were in the middle of the very beginning to the present time, no question.
So I'd also like to pose that same question to Dr. Rosenblatt to share, your comments and perspectives on where we were, where we're going, and what you think the future holds.
Thank you, Chuck. And Chuck, thank you for the opportunity in participating in what I thought was a really excellent 2 days in which I learned a great deal from all the speakers. I can comment mostly on how the advances have really moved forward in the area of diagnostics, whereas starting really with the pioneering work in Leon Rosenberg's lab and the assignment of the first complementation groups of A, B, and C. The somatic cell complementation is really quite difficult, and where you can make a preliminary diagnosis, you'd have a patient who may have a particular phenotype. Actually characterizing them was not that easy. And today, because the genes have been discovered, you can do a panel, and if you have metabolites elevated and you have pathogenic variants identified, then you can be pretty sure of your diagnosis and at least characterize your patients properly. My concern, as always, and Harvey can probably, maybe if there's time, go back to this, is the early days of amino acid analysis, of labeling people with disease who don't have disease. So I'm very concerned that diagnosis is made on the basis of molecular panels without really validation that's there. And I'm very concerned, and I was interested in the comments on our clinical validity and recommendations for newborn screening. Especially we saw with the data from the California screening program that many of the variants that came up in cobalamin C were really late onset alleles. And so what happens when you institute newborn screening for what is really adult onset disease in the majority of cases, and how do you approach that? So I think the opportunities are great. The technologies are great. The opportunities for those who are open to it for pre-implantation genetic diagnosis is also great. So I'm very excited about the future, but also cautious, particularly about what I heard yesterday about the promises we make in terms of long-term outcomes and our ability to deliver. So we're very good at diagnosing and viability, but can we maintain the patients under good care? Also, the increasing costs of care, and how we can, it's one thing to make promises and put in therapeutics, but how do you get it to the entire world in a way that it can be afforded by the patients who need it?
Thank you. Those are important lessons we always must, take forward with, especially with screening, which is the next segue, which I'm going to ask Dr. Pasquale to comment on, and then the last comments will be reserved for Dr. Human. So with that as a segue to what I was hoping you could comment on, with respect to newborn screening and the comments we just heard, what going with the implementation of rapid genomes?
Sorry, Chuck, you cut off a little bit. Can you repeat?
Okay. Yeah, I will. I was going to ask you to comment on where you think the field is going with respect to newborn screening as we consider the new genomic technologies like rapid exomes, rapid genome sequencing, and targeted identification of genes where we may have a metabolic perturbation associated with it. I think the question of what do you do with the whole genome, beyond newborn screening is something separate, big discussion. But I'm curious if you could share that perspective for us, because as was just mentioned, the experiences with the exome sequencing following up on newborn screening, does open this an age-old problem of finding variants that are they disease causing, and what do we do to treat them? But then again, finding patients that have intronic variants in genes where we would miss them otherwise. Maybe their metabolic perturbations aren't always present in some patients. For example, cobalamin C patients can have low methionine, low C3, and they can be missed. And I've seen this in my own personal experience multiple times, and they're severely affected patients. So clearly we want to find every patient. But with that, could you make some comments on your perspectives and your opinion about where this field stands and where it's going to go?
Well, my perspective, of course, is of a biochemical geneticist. So I give a lot of weight to functional evidence. And but having said that, I think that where everybody's going is really, or they're going to try to go, is to integrate. At least I hope they're going to try to integrate the functional data with the molecular data. It's to me that would be a good approach to that. What are the barriers for to implementing exome sequencing or genome sequencing at the level of newborn screening? Well, you have, of course, cost. That's not insignificant. You have turnaround time that needs to be considered, and also one of the, I don't know if it's a barrier, but it's a challenge, would be interpretation of the data, that you need expertise built in there to interpret the data, and especially to integrate the molecular data to the metabolic and to the functional data. I think that it can be used right now in our states. They're doing a research project, and they're using it even as a second-year testing in a way. Again, how do you do that? And assuring also that patients in the family are informed of what this type of analysis is going to uncover. That's another issue that I think we need to think about when we do this type of testing. Regarding those late onset diseases, that's a big question. We are already doing that, right? We're screening for diseases that do not that have a late onset, and are creating some interesting challenges.
So it's fair to say we're not there yet.
We're not there.
The promise is there, but that's, again, an excellent segue to what I was hoping Dr. Humer could comment on, which is, now that the patients have identified, we found them, you talked about the EHOD databases, which are really an amazing resource. And I'm wondering if you could provide some comments and perspectives on, what should we be doing with the patient populations we find, and what's sort of maybe some comments on organizational approaches. I've been to Europe many times. I'm sort of, to myself, I think, when I think of a regulatory burden of, just between centers in the US, I sort of wonder in Europe how that could even happen with so many different countries and everybody having independent opinions. But then also, if you could comment as well on, sort of your opinions of what are some of the exciting new treatments that you're working on. I noticed from your EHOD, you talked about the high-dose hydroxycobalamin treatment. And so maybe if you could make a few comments about that. And then the last few minutes, we reserve open to comments for those who are still with us with their cameras on that might want to make a comment. So please take it away.
Yeah, thank you. Yeah, you're right. It's a bit of a burden to keep all those people together all over Europe and extra European countries. But I think it's the enthusiasm for the project that drives people together. And so everybody is doing a tremendous job. And so we're getting along quite well at the time. And I think the chance in this project is that we come up to a unified perspective in a way, because Regarding, for instance, the new therapy, high dose therapy for CBLC, I think we can only make this happen in Europe if we go together, because there's no drug. We are ready, we have a protocol, we have a lot of ideas how we could study the effect of this high dose treatment in a standardized and effective manner, but there's no drug. And I think the only way to approach this is on a European basis, to keep all the countries together and go to the regulatory bodies and try to make that happen. And I think that's
I was going to pose a question to you, which is, if the different countries don't have the same newborn screening panels, could that complicate such trials? You know, for example, in the U.S. we have a uniform panel that most states have adopted, but I think in some European countries you're not screening for, say, cobalamin C deficiency.
Yeah,
please correct me if I'm wrong.
No, that's absolutely correct. I'm working in Austria and in Switzerland. In Austria, we do screen. In Switzerland, we don't. And you have as many countries, you have as many newborn screening panels in Europe. There's no unified panel until now. There are great plans, but nothing happened yet. So yeah, that's a problem. But I think we have some core countries that have it's quite similar panels, and we can start from there and then try to convince other people to join us. I'm sure that we should screen for CBS, we should screen for MTHFR, for the cobalamin-associated disorders, because we can really make lives better. And yeah, but as you know, in screening programs and panels are always also political decision. It's a matter of culture, it's a matter of how your state is run. And so, yeah, we are starting, we are being modest and just start to go with very small steps and hope we can just bring more people in. Yeah.
Well, certainly, I think your efforts could help newborn screening get implemented more widely in Europe as a large benefit.
It is implemented in all states, but it's handled very, very differently. Yes, and we're working on this.
Yes. Well, I meant expanded newborn screening in the sense of, you know, the long list of disorders that it's constantly growing in the U.S.
It is, yeah.
Yes, well, we're coming to the end of our panel discussion, and I did want to open the floor to members of the group here that are still with us, that are still paying attention, that are still engaged in the conversation here. That would be, you know, basically anybody who's on the line as a panelist who may want to make a comment or you know, offer a closing remark. I would ask that it would be short, but please, I want to extend that to the group to see if we have any takers, and if not, we will adjourn in a minute or two.
I can't resist. So the costs here, you know, I recently got my genome sequenced. It's come down considerably. It was $299. Now, maybe that was a loss leader, but you know, when you're talking $299 versus $125, I just feel like, you know, this is going to happen, and it's going to behoove us to figure out what to do with that information, and how should that replace newborn screening? Should it be the tier 1 screen that you use to decide what to do? I just think these are the issues that, you know, as a community, and I'm not a clinician, so I just can speculate, but I do think it's something that is coming soon, real soon. And, you know, I can tell you from my own experience, I have a weird proline hydroxylase mutant that I'm not homozygous, but I have 2 what appear to be functional alleles. Is that responsible for my back problems? Who knows? But, you know, I don't know. What do you do with that information? I have no idea. So I just bring it up as something that I don't know. I somehow we need to have a scientific way to determine what's the most cost-effective way to find these early patients that are intervenable, and whether it's one way or the other, and to think about that. Because I think we're all pretty sure, if you've got 2 null alleles of cobalamin C, you're going to have a problem,
Yeah, that's a great food for thought as we move to maybe our next person that would want to offer a remark. I see Ken, you're next on my screen, so if you would like to say something.
Chuck, I just would like to make a comment. I appreciate, the comments of Martina, because it's true that in Europe, our policy for newborn screening is extremely exogenous. Actually, for instance, in France, we are starting with homocystinuria, and we are not discussing cobalamin C, just to show you how delayed we are, really. So she's completely true, and we should have really a common policy. And thank you, Martina, for your efforts.
Anybody else? A few people are.
I don't know if I can talk now.
Yeah. Your hands up.
My good friend Warren kind of made the argument on why it's inevitable, and then he undercut the argument on saying why we're not even close to being ready to implement genetics, DNA as a screen. So I would just echo his last point, is that we really need some science, and research so far has said it's not better than what we're doing now. And certainly the US has no great claim to homogeneity in newborn screening, as lots of the procedures and screens and follow-ups differ from states, and there's more US states than there, I think, are countries in the EU. So it is a call really for a lot more work in this area and true demonstration projects.
And I would like to say, yes, that's absolutely correct. I think the fact that it's gonna come, is gonna be here, and it's gonna happen, doesn't mean though that we are ready. And perhaps if the experience that we have had so far with newborn screening has taught us something, is that we need to be proactive in finding ways to make this happen the right way.
And I just add one quick thing. The cautionary tale from non-invasive prenatal screening is that if industry decides this is something everybody should have, they're pretty hard to combat with data. And I do worry right now, I don't know about in Europe, but in the US, the states take care of the newborn screening. It's not a profit generator for the most part. It's done economically and efficiently. If somehow the companies decide that every newborn should be sequenced, and that's the best way, and they go to Capitol Hill to do it, their voice can actually go a lot further sometimes than the scientific facts. So I am concerned about that.
And I'm very concerned about that. And there were some people who showed the growth of direct-to-consumer testing. We have a large patient group, and it's increasing that your patients are coming to you with variants that are actually flooding the clinical requirements of patients with illnesses that you have to deal with, and the resources aren't there clinically to deal with it. So I think we can't ignore the direct-for-consumer. So we have to find a way to get at that data and to have an influence on policy, because that's out of the bag, that's happening, and at large numbers. So it's there. It's just a matter of how we deal with it.
Well, we're 5 minutes over. The discussion, I think we could keep going on and on. It's really sort of an exciting, for me, this has been sort of really super exciting to, to have this discussion with everybody. But I'm going to close the meeting now. And again, thank everybody for the participation, and also, again, acknowledge the wonderful AV support we've had from the NHGRI. And to say again to Dr. Manoli, Sloan, and Shalachkov, they really did most of the organizing. And so really, it's been an amazing event. And thanks again to everybody. I look forward to seeing you all someday soon, I hope. I remain optimistic that it won't be that long. So with that, good evening, good afternoon,
Thank you.
everybody.
Thank you all so much.
Bye, everybody. Yeah, great meeting.
Bye.
Thank you for joining.